I've given us enough time now. I think our numbers are slowing down. So I will officially start the webinar. Welcome, everybody. This is a joint session between ISPG and PGC, the Worldwide Lab on Longitudinal Approaches in Psychiatric Genomics. I'm Jessica Dennis. I'm an assistant professor in medical genetics at the University of British Columbia in Vancouver. I'm going to be co-chairing the session along with Maria Coromina, who's an instructor at Mount Sinai in the departments of psychiatry and artificial intelligence and human health. Before I introduce our speakers, I wanted to give a little bit of preamble about the session. I'm so happy to see so many of you here. As I said, our numbers are stabilizing, and we'll take a look at our final numbers. I know you all can't see how many people are here, but trust me when I say that we've got a good audience today. And I'm excited to see so many of you here because it means that you also think that we need to be doing more with longitudinal data in psychiatric genomics. And this is something I think about a lot, and along with some wonderful co-authors, we actually have a review that's in press in biological psychiatry, where we argue that longitudinal data analysis is going to be instrumental in advancing psychiatric genetics. Now, the good news is that we also think that we can do a lot with the data that we have in hand. This is a figure that was generated by the second author on this review article, Haley Thorpe. And what she did was she dug into the supplement of the most recent PGC PTSD GWAS and pulled the average age of cases and controls, which was in the supplement. And she plotted the average age of those cases and controls. You can see along the x-axis there for each of the study samples that were in the GWAS meta-analysis. And you can see that even though a lot of the data are coming from middle age or older adults, there's a fair bit of spread in the data that we already have in hand. So this tells us that there's opportunities to leverage age or time-related information in existing data sets. Another theme that we explored in this review is highlighting results from emerging longitudinal GWAS that are showing the potential lost information by focusing on binary case control definitions, which is what we've relied on in GWAS and psychiatric genetics for a long time. I think getting at that potential lost information is what brings us all together today, learning about some different approaches in psychiatric genetics where we can harness this longitudinal data. So with that in mind, I will formally introduce our speaker. Well, actually, before I introduce our first speaker, I will just do some housekeeping for the webinar. The format, we will have 3 15-minute talks. Please put your questions in the Q&A box. At the end, we will have a Q&A session that will cover all 3 talks. So we can either read out questions that are in the Q&A, or we might prompt you to unmute yourself and ask the question yourself. Our first speaker is Hongtu Zhu from the University of North Carolina at Chapel Hill. Hongtu Zhu is the Keenan Distinguished Professor of Biostatistics at the University of North Carolina at Chapel Hill, with joint appointments in statistics, computer science, and biomedical imaging-related programs. His research focuses on statistical learning, imaging genetics, biostatistics, AI, and large-scale biomedical data analysis, with broad applications to brain, heart, genetics, and complex diseases. Hongtu, I will stop sharing and let you take it away.
Thank you for a nice introduction. Let me see. Can you see my screen? I needed to how do I go to the presentation mode?
I usually use on the bottom right of your screen, the little projector.
This one?
Yes.
Okay. Good morning, everyone. It's my great pleasure to be here, and also thanks organizers for the invitation. Today's talk is about the genetic architecture of disease timing. Okay. And this is basically just based on the joint work with my students and also my collaborators, in particular Patrick. And this is basically an overview the method. And actually, this is basically our method called the AFT-LDSC method. And also, this is the kinds of the key pipelines. And also, we applied this method to UK Biobank. And all these some of the key findings associated with basically our tools. Okay, and uh, and this is the introduction. And actually, the current challenges, the timing of the disease onset and the rate of the of clinical progression are fundamental feature of the human biology. And what are the most genetic analysis as see basic disease risk? There's a lot of tools associated with high efficient GWAS for quantitative and also binary traits. Okay. The typically people collapse basically reach longitudinal trajectory into the static case control indicators, the well-developed LDSC tools for uncovering genetic architectures. But actually, this all these most of these large scale biobank data sets they do contain the basic disease timing. Okay, and one critical question is how to integrate decades of time-stepped EHR records into the basically downstream the GWAS analysis. And actually, but there's a major challenge associated with heavily based. Most of these is basically as EHR basically survival traits are heavily censored event time. Okay, there's no popular tools like LDSC. And our basically contribution is basically try to develop tools for the time to event phenotype. Okay, and they also some of the existing tools for time to event phenotype. Basically, most of these tools are listed here are powerful and efficient GWAS for locus discovery on true event time. And all relying on the hazard models for frailty basic models. The summary statistics cannot be applied by LDLC. And actually, there's some of this major limitation of this method. Basically, there's no definition for LD, LP, and sleep heritability for survival traits. There's no patterns for genetic architectures on the disease progression, and also no analytical tools for LDLC style to develop for sensitive outcomes. This is basically the key motivation for our student project. Basically, we want to basically analytical tools for time to events basic phenotype. Okay, and this is basically our tools fill the gaps. Basically, well, these are the key basically four key modules. We're showing the accelerated phenotype models for TTE phenotype, and also we define this censored aware the summary statistics and censored censoring adjustment factors. And also we establish heritability, annotation enrichment, and a genetic correlation for survival traits. And also for the first time, we will the genetic patterns of disease progression through the analyzing the UK Biobank data. And these are basically the key methods we have. And this is basically the notation for observed data. You have the genetic basic vectors. Y is censored basically the observed times, and the TI is basically event time. Basically, CI is the centering time, and the data is indicators. And we feed it basically accelerated basic phenotype basic models. Is the typical models in survival analysis. We consider log TI equal to this. And also we can define these sleep heritabilities on the time to event scales. And basically, these are typical assumptions people are assumed for quantitative traits. And also we define these heritabilities equal to this, the ratio of this. This is basically our definition of the sleep heritability. And also we can define the genetic correlations and on the time to event scales. And suppose you have two cohort and actually you have two time survival outcomes, you assume the similar models. And then you can define the variance of this and this is the final basic genetic correlation between the two time to event traits. Okay. You can also basically, we can replace one of these basic equations for the binary trace and also for the quantitative trace, okay? This is basically the key formulation behind it and the settings. And actually, we define a summary statistics. And actually, the key problems as under the censoring is basically true event time is hidden for censoring individuals, okay? And also the Cox summary statistics is not meaningful on these variance scales. There's no current efficient basic survival GWAS mod method under the AFT. And actually in our the results, and actually there are two key kinds of statistics. One is the censoring aware the summary statistics, and actually it's defined by this. And this is and also we have the censoring adjustment factors, and it's defined by this. Okay, these are the two are very important for calculating the downstream heritabilities and also genetic uh, correlations. And this is basically on the left panels. I listed basically the key formulas for standard LDLC. Basically this is summary statistics. And this is heritabilities, these equations. The slope estimate of the is a SNP basically estimate the SNP heritabilities and intercept captured confounding. And also the genetic correlations. And actually this is the basic formulas. And NS is the size of overlapping individuals, slope estimate the genetic correlation. These tools has been widely used in different basic studies. And actually, you see that basically we parallel to this, we define this AFT-LDSC. Basically, our summary statistics is given by this. And also this is the adjustment basic factors. We can show that the heritability for expected value of the VJ squares is equal to this. And actually, we have this basically, you see that this is very similar. We can still basically using regression to calculate basically this. And also genetic correlation is given by this, okay? And this is basically is fundamentally important basically for all these downstream calculations. We spend a lot of time to figure out how to do all these calculations. And also we can do this basic adaptation enrichment. And also basically we can, similar to the expression basically LDLC, we derive these formulations. And also this TOV is annotation specific post-SNP heritabilities. And also this is regression weights. And also we use the weighted regressions, and we follow the LDLC. We did this kinds of calculations, and this is basically we also have these iterative the weighted regressions and to do the to calculate the optimal weights, and also we can calculate the standard error estimations by using Bootstrap. And this is the practical workflows before the after LDLC. Your first one, you need to pick the reference panels, LD panels. The second step, you need to remove the covariance effects. You want to adjust it. For most of our cases, basically, we correct for sex and plus the genetic PCs. And also, you need then you can calculate the summary statistics. You need to standardize basically on all the y and the xi, and also you did the calculations. Then you now after you did this apply this step 1 to 3 to your data, and now you can apply our AFT-LDSC. And this is basically we show the simulation studies to validate our tools. And this is basic genetic data, and also this is the basic causal MF and the LD related architectures. And also these survival time distributions. And we are basically consider different basic survival distributions, the standardized normal, logistic, the Gumbel distribution, and also the censoring time. Basically, we consider two settings. And then you see that for some settings, there's no censoring, but also for some extreme cases, there is censoring, okay? And also these LD panels, and also we can calculate the binary basic outcomes. And this is basically simulation results. Basically, you see that for our when gamma equal to 0, our method exhibits a minimum bias even when sensor rate is extremely high. And then when gamma equal to 1, basically our method exhibits a downward bias that is scaled with the sensory. But the traditional basically liability scale, the LDLC, has the similar trend. Okay, this is the basic left is AFT-LDSC, the right is LDLC's liabilities. And also we consider different MF, and also you see that our method is basically when MF basically from the 0.2 to 0.5, our method basically yields the lowest bias and variance. And also we did the following basically additional simulation for enrichment and genetic correlations. And actually, you'll see that basically our method is pretty basically unbiased. And in most of the cases, even for extremely basically censoring rate, you'll see that our results showing that. Even, people, this is basically extreme challenging cases. And also did additional some distinguishing points. And actually, this additional results. After basically LDLC exhibits substantial smaller bias then the liability basically scaled LDLC, okay, on the distribution misspecifications, okay. And with the same data, and actually after LDLC and LDLC can yield divergent heritabilities and the genetic correlation basically and estimators. This is very important. Basically, for many of the previous studies, people are just clumping the data into the binary outcomes. Okay, basically, our simulation show that basically if you using AFT-LDSCs, basically the traditional liabilities, they could get the different results. Okay. And basically, the take-home message is our tools basically preserve the reliability and the efficiency of the LDLC framework across a wide range of settings. Okay. And this slide shows you some of the results. Basically, it's the UK Biobank Phenotypes the Atlas. And actually, we can see the 508, the time to event phenotype. And also, this is the mean censoring rate, extremely high, okay? Because it's older population, and this is basically, and also we figure basically choose the appropriate basically FT models. Basically, we do model selections, and also basically the mean survival times and 60 years older. And this is basically the total heritabilities. And actually, this is the 508. And then this is basically there's FDR, basically, there's significant to 60s. Banfonic insignificant to 137. And this score is greater than 5. For most of the cases, the survival basically time to event heritability is larger than liability basic heritabilities. For some mental and circulatory disease, this is a reverse. And also, this we did further did the functional architectures, and basically this share the genetic basis between survival and the binary scales. This is basically this is the figure in the panel A, and also the progressions and sensitivity based biologies, and you show that our tools basically show the preferential enrichment in all these regions. And also cell type grouping and also enrichment patterns are largely consistent for traits dominated by a single tissue systems. And for multi-basic systematic basically traits, enrichment are generally more pronounced than in binary phenotype. Okay. And also we calculate the genetic correlation between the TTE and all the binary phenotypes. And the nearly identical basically global polygenic architectures. The genetic correlation between each of the survival phenotype and the corresponding binary counterpart are consistent near the unit. Okay, but an FT-based LDSC revealed the progression relevant to basically polyatrophy. Okay. And also, we also calculate the genetic correlations with the imaging traits, and also basically between the survival traits and all the imaging traits. And then we consider basically the wide matter microstructures and the regional brain volumes, and also the cardiovascular features. Okay, this is basically allowed us to link the basically imaging traits, the organ traits with the survival traits. And the conclusion I want to make, basically, the methodological, we provide a natural linear scale definition of the sleep heritability and genetic correlations. And we introduced allowable summary statistics that capture the correlation between the observed outcome and the genetic variance. This statistically effectively generalize LDLC framework to censor survival data, and also enable basically same the analytical breadth as the standard LDLC. And biologically, basically, we assume basically the broader concordance between the risk and the progression, and also show basically high resolution signals from disease timing. This is quite exciting because you see that we can handle extremely basically sensor rate, and also we are sure basically the environmental dilutions of the progression genetics. And this is basically this is some additional basically discussion regarding to recent conflict reporting on the disease progressions, and also several limitations of our method. But in general, basically, we provide tools that bridge the non-standing gap between the disease risk and the progression. And our tools offer a powerful tools of shifting the focus of the genomic medicines from the static basic risk assessment to a dynamic understanding of the human health span. This is basically the open source basic software packages, and you're interested, basically you can basically download it, and also the summary statistics for AFT-LDSC and the summary statistics for binary imaging for all the imaging traits. And also this is basically we develop the tools basically from this static disease risk to dynamic health genetics. This is some of this summary of my talk. That's the end of my talk. This is some of the key references. Thank you. I stop here.
Thank you so much, Hongtu. I'm going to pass it over to Maria to introduce our next speakers.
So it's my pleasure to introduce Dr. Noam Beckmann for the second talk of our webinar today. Dr. Beckmann is an assistant professor at the Icahn School of Medicine, where he is leading research at the intersection of AI, digital medicine, and neurodegenerative disease biology. His work does integrate a lot of large-scale genetic, transcriptomic, proteomic, and a variety of omics data. And with no further ado, Noam, please take it away.
Thank you. Can everybody see my slides? Yes, okay. Thank you for inviting me and giving me the opportunity to present some of our latest work. And today, I'm going to be talking about how we leverage the temporal components present in sparse longitudinal data to try to understand the genetic control of Alzheimer's disease progression and to establish a novel paradigm for disease risk. And of course, I can't switch slides. There we go. So why is this important? Well, if we take a bunch of individuals in a study at time 0 and we look at their case control status with Alzheimer's disease, many of them may not have Alzheimer's disease. But if we take the same individual sometime later, many of them may have Alzheimer's disease. And that really emphasizes the fact that it's very important to consider not just the state at the specific time, but rather how disease progresses over time. And I would like to make a postulate here that I believe that every disease progresses along a timeline. And what my lab is doing is that we leverage the temporal information contained in these sparse longitudinal data to reconstruct disease timelines. I emphasize here the word timeline because this is a word that I'm going to be consistently using throughout the rest of the talk. And now I'm going to only give you one slide on methods because I don't want to take the focus away from the results. And this is a method that was developed by Ryan Thompson in my lab that we call timeline inference method. And I'm not going to tell you how it works right this second for the sake of time, but I will highlight what I believe are key advantages of this method relative to what exists out there. The first advantage is that we require very minimal input. The only things that we need to do to be able to build a timeline are the relative time link within individual ID, and the data itself. We also make very minimal assumptions here. We do not, the only assumption that we're making is that time progresses monotonically, but not necessarily linearly, and that some features in our data have a temporal signature. And finally, the only other assumption that we're making here is that all individuals are on the same timelines for our disease of interest. The advantage of this design is that it doesn't require any knowledge of known biomarkers of disease or milestone or anything like that. It does not assume equal progression rates across individuals, and it can scale to hundreds of thousands of samples and features. And maybe most importantly, as part of this framework, we've also developed an unsupervised and accurate estimate of the timeline inference performance. And this is really important because as far as I'm aware, there's no method out there that allows you to do that. And if I tell you, yes, I can build a timeline, but my timeline is random, what's the point? I need to know whether my timeline is random or not. And this is really what we try to achieve here. So just to give you some analogy of what this means, every one of my analogies is about cars, and I apologize. Imagine for a second that each patient is a car, each individual is a car. Our goal here is to build a timeline of the disease, so that's akin to the road on which the car is driving. And that's really important because once you know this timeline, you can learn properties of the road. For example, here there's a very sharp left turn at the beginning, and that might be a place where you'd want to modulate the speed of the car. Maybe you want to put a speed limit. Maybe you want to put a red light, a stop sign to avoid an accident. But it also allows you to learn properties of the car. For example, how fast is the car driving on that road? And that is only definable once you've defined the road. That was the only slide on a method, and now let's move on to the data. And this is work that was led by Eli, a fantastic graduate student in the lab, and Daniel, one of my closest collaborators, where we leverage this absolutely incredible resource that's been put out there by the Alzheimer's Disease Neuroimaging Initiative and Alzheimer's Disease Metabolomic Consortium, where they generated this incredible resource. They followed patients for many, many years, collected samples throughout, generated all kinds of clinical metrics, and then generated molecular data at multiple time points from these patients. And the specific data sets that I'm going to be talking about today are the different lipidomics and metabolomics data that were available in this cohort that had a sparse longitudinal component. So as I mentioned in 2 slides ago, I believe, I told you we can estimate performance of timeline inference. So the first thing that we wanted to do here is ask, can we even build a timeline of Alzheimer's disease from this data? And if so, how well are we doing? And so what I'm showing you here is the performance estimation for the 5 timelines that we built, one for each modality of data in this cohort on the x-axis, and on the y-axis you have the performance estimation tau timeline. And just to give you some intuition on what that means, a performance of 0 would mean random performance. We are absolutely terrible at building a timeline. A performance of 1 is a perfect performance. And performance of 90% accuracy would be equivalent to an estimated performance of 0.8. And you can see here that for serum metabolites, we're actually not that far from that. So we are indeed able to construct timelines of Alzheimer's disease that our performance estimation tell us represent the true underlying timeline of Alzheimer's disease. Now, that's not quite enough. Now, because we have all these different modalities of data from which we infer timelines, one of the really nice things about the Alzheimer's Disease Neuroimaging Initiative is that they generated the data across all these modalities for virtually the same individuals at the same time point. And that gives us really this fantastic opportunity to ask, are we actually recapitulating the same disease timeline across these modalities of data? And if we aren't, then maybe we're not actually learning the timeline of Alzheimer's disease. And so what we did is we simply performed Spearman correlation between these timelines. And what I'm showing you here is a heat map of the Spearman correlation across the different timelines built from these different modalities. And suffice it to say that all these modalities of modalities of data-derived timelines recapitulate each other very significantly. The worst p-value here is about 10 to the minus 30. So we're doing fairly well. And underlying that, we are indeed able to recapitulate the same timeline of Alzheimer's disease across these different modalities of data. At this point, we have some confidence that this timeline is actually representative of the true timeline of disease, but that's still not quite enough to convince me that this timeline is good. And in order to convince me that this timeline is good, what we needed was to spend some time and try to figure out now, is this timeline actually predictive of clinical decline, of clinical presentation of Alzheimer's disease? And so what we did is we simply asked, is the temporal location along these timelines for my cohort predictive of dementia score? And in this case, I'm only showing you clinical dementia rating sum of boxes, but we've done that across every possible dementia score that were available in ADNI. And what I'm showing you here on this plot is on the x-axis, the estimate of the association while accounting for age at sampling. On the y-axis, the different modalities of data that were used to build the timeline, and the colors represent the group of individuals that were used to do this prediction. And what you can see here is that using only the temporal location along the timeline, we are able to very significantly predict what is the CDR-SB score for the individual in this cohort, specifically for Alzheimer's disease. And that's really nice because now we know that our timelines recapitulate clinical progression of the disease. But that's still not quite enough. As many of you know, Alzheimer's disease is also a disease of biomarkers. There's a bunch of biomarkers that are known to be associated to the disease that are mostly used for diagnosis, but have been postulated to also be important for progression. And so what we did is we, again, asked the same question: can our temporal location along the timeline predict the level of these plasma biomarkers that have been previously associated to AD? And here I'm showing you only the plasma A-beta-40 score that we can show is specifically, again, associated to temporal location along the timeline in the AD group. And we've done that for an array of other ones. But suffice it to say here that plasma A-beta-40 is only one of two components that are used for diagnosis of Alzheimer's disease, which is the A-beta-42 to 40 ratio. And the ratio itself is not associated to temporal location, but each one of the components of that ratio was, which is emphasizing that it is maybe possible to use this timeline to identify novel biomarkers of disease progression. But that's still not quite enough to convince me that this is actually representative of time. Like the word timeline has time in there. It's very important to show that what we're showing here is not just severity or something else, but it's actually time. And the only temporal milestone that we know for individuals with Alzheimer's disease is how much time has passed or is going to be until they have a diagnosis. And so what we ask is, can we predict time of diagnosis using only our timelines? And what I'm showing you here is that, yes, indeed, we can, very significantly so. The R-square is actually pretty good. And the point that I would like to really emphasize here is that this prediction is done completely independently from any biomarkers of Alzheimer's disease. And what this means is that we have an opportunity here to potentially drastically increase the performance of how we predict age of onset of Alzheimer's disease by integrating this timeline-derived knowledge with the biomarkers of Alzheimer's disease. And so at this point, hopefully I've convinced you that our road is actually representative of the road on which the patients with Alzheimer's disease are driving. And so I'm going to switch gears a little bit and start talking to you about the properties of the car. And the property of the car that I would like to focus on today is how fast is that car driving on the road? In other words, what the progression rate for people with Alzheimer's disease. And one of the beauty of having longitudinal data is that once you do have longitudinal data and once you do have a timeline, it is very simple to define progression rate as simply the slope of the relationship between the real time and the inferred time elapsed on the disease timeline for each individual. And now that we have this individual specific metric, we wanted to ask, is this metric under genetic control? And so we run a GWAS here to try to see whether there was any association between Alzheimer's disease progression rate and genetic loci. And I'm showing you here only one Manhattan plot. We've done this across every modality of data, but for the sake of time, I'm only going to show you this one. And what we were very happy to see is that despite our fairly low sample size, there's only about 1,300 individuals in this cohort, we get a well-behaved GWAS where we can identify not only genome-wide significant loci, but also a bunch of suggestive loci. And what's interesting is that when we look at the mapped genes here, these are not genes that we've canonically seen as associated with AD genetics, but most of these have been shown to be important to the etiology of Alzheimer's disease in other contexts. And so because of our sample size, it's really important to think about, do this signal replicate in larger outside GWAS? Now, there is no existing GWAS of progression rate because nobody's defined progression rate before in the context of Alzheimer's disease. But because progression rate, we assume, is not entirely independent of case control ascertainment, we ask, can we leverage these very large Alzheimer's disease GWASs out there to replicate our finding? And what we see here, what I'm showing you here on this plot, is that yes, indeed, we can replicate our suggestive loci at nominal significance more often than expected by random chance in these external GWASs. And now I'm going to bring back this Manhattan plot that I showed you a minute ago to point out something specific that may have eluded those of you that are not used to looking at Manhattan plots for Alzheimer's disease. There is something here that's missing, something that I've personally seen on every single Manhattan plot that has ever been shown for Alzheimer's disease, and that should be on chromosome 19. There is no peak whatsoever at the ApoE locus. And this is really important because when we think about ApoE, it's the strongest genetic risk factor for Alzheimer's disease. So what is going on there? And this led us to ask, are our genetic association with Alzheimer's disease progression rate independent from the ApoE genotype? And so what we did is that we performed the exact same GWASs, this time controlling for the APOE genotype. And what I'm showing you here on the left with p-value distribution and on the right with effect size is that accounting for APOE does not meaningfully change the results of our GWASs. And that was true across every single one of the modality from which we derive timeline, showing that our genetic associations here are truly independent from, at the very least, the strongest risk factor for Alzheimer's disease. Now, going beyond this, we wanted to ask, what is the causal relationship between progression rate and clinical phenotyping of Alzheimer's disease, in this case, case control analysis? So what we did is we run a one sample Mendelian randomization analysis. And what I'm showing you here is that progression rate is indeed significantly observed as causally upstream to case control differences across every single way we define the progression rate, across every single modality of data. This is also true for clinical dementia rate, sum of boxes, so cognitive score at baseline, as well as progression of cognitive score over time. And that really emphasizes that progression rate is really an important phenotype that we need to think about when we try to understand what's going on in patients with Alzheimer's disease and how we're going to modulate their disease, etc. And at this point, I've shown you all these results, and we have these nice genetic associations, and it's independent from APOE, and it's causally upstream of clinical phenotype. And this led us to try to think, how do we reconcile this with the way we standardly think of case control risk of developing disease? And so what I'd like to propose here is that maybe we would like to augment the way we think about AD susceptibility in general, or AD risk, and propose a novel model for total AD risk that is some function of the risk for AD potential and the risk for AD progression. I'm going to define that for you in a second. When we think of susceptibility to a disease, historically, what we think about is this one-dimensional axis where you have your susceptibility to Alzheimer's disease in this case. And if you have a high risk, you probably will develop AD at some point in your lifetime. If you have a low risk, you probably won't. And that's very nice, and that's certainly real, but I think it's an oversimplification of reality. When in reality, we should have multiple axes, this risk for AD potential, which APOE would likely be contributing to, and the risk for the rate of AD progression that is captured by the GWASs that I've shown you today. And if we were to take every individual in the population and project them onto this plane, we would end up with some subdivision like this, where on the upper left quadrant, you have the people that have a high risk for rate of AD progression and for AD potential. These are the people that are most likely to develop AD in their lifetime. On the lower left corner, you have the people that have a low risk for both, that are least likely to develop AD in their lifetime. And when we think about the way we currently define susceptibility to AD based on case control GWASs, what we're measuring is one number, and what that number represents is any combination of these two independent risks that are represented by this line on the plot. And so really, if we want to understand more the biology of Alzheimer's disease, we need to be able to distinguish these two risk factors, and this is something that our method allows us to do. In order to see whether the data supported that this model is actually real, we simply ask, can we better predict age of disease onset using both the case control polygenic risk score and the progression rate polygenic risk score than the case control polygenic risk score alone? And what I'm showing you here is that in the ANI cohort, yes, it is the case, we can significantly do so. And we replicated that in a much larger cohort, the Alzheimer's disease sequencing project, where they have about 10,000 individuals for which we have genotype data and age of onset. And again, we can show that the addition of the progression rate PRS provides meaningful information that allows us to better predict age of disease onset, supporting this multidimensional paradigm of disease risk. And so in conclusion, hopefully I've shown you that we are able to build timelines that recapitulate clinical disease severity, and that timeline progression rate reflect a distinct biologically meaningful dimension of AD. We've also identified novel genetic loci associated with this progression rate that are largely distinct from the way we generally think of case control differences, including APOE. And I've shown you that this data supports a new multidimensional model for AD risk. And I think this provides also a framework for studying AD and any other progressive disease through not only the lengths of static case control differences, but also the lengths of disease dynamics. And I have one acknowledgement slide here. I'm a little bit over time, so I'm going to go fast. Just want to name Eli and Daniel that did most of the Alzheimer's disease analysis, Ryan that built the method. And we are standing here on the shoulders of giants, the people that have generated these absolutely incredible resources that allow us to even do this science, such as the ADMC, ADNI, and ADSP. And thank you for your attention.
Amazing. Please, everyone, stay for the discussion. We have one more excellent speaker. So I'm very happy to introduce Dr. Anina Lod. She's a postdoctoral fellow here in Mount Sinai, and her research focuses on understanding how different molecular processes can shape the human brain across health, aging, and neurodegenerative disease. And Anina, the stage is yours.
Thank you. You guys can hear me all right, right?
Yep, well, great.
Amazing. Okay, so I'd like to thank the organizers for this opportunity to present this work. As you guys can probably tell by this title of this project, this is still very much a work in progress. It is very hard to come up with a concise title when you're still just piecing through results. But yes, I'm very excited to share this work. So you all have just heard about the lab's interest. I'm a postdoc in Noam's lab, and we're very interested in understanding the time component of disease. Noam talked to you about the work that we've done in the context of Alzheimer's, and I'm excited today to share the work that we're doing in the context of Parkinson's disease. Now, before I dive into some of our findings, I just wanted to start with a reason as to why it's so important to understand the timeline of PD. So we start with the clinical diagnosis of PD. Now, across individuals, as you can imagine, the age of onset can vary, right? When people are actually diagnosed, they can be different ages. Their symptom profiles can also vary. So PD is a multi-system disorder where you have both motor symptoms and non-motor symptoms. So motor symptoms, things like tremor and rigidity, non-motor symptoms, things like sleep deprivation and cognitive effects. And so these different profiles can present in many different ways across individuals. How the disease progresses can vary across individuals. So some people progress quickly, some less quick, and the genetic background across individuals can also vary. To make this even more complex, we also know that there is this prodromal period, which essentially means that this age of onset can be a lot earlier than the age of a formal diagnosis. Generally in this prodromal period, we see non-motor symptoms that are presenting, which could be a reason why you're not picking up, or they're just generally not being picked up earlier with clinical diagnoses. And so in short, what I've shown you here is that PD is a heterogeneous, multi-system progressive disease. And so as you can imagine, studying all these pieces can be incredibly challenging, especially without really understanding the actual underlying timeline of the disease. And so that's what I'm going to be talking to you about today, how we're working on building this timeline of PD that can hopefully start aligning all these pieces together to better understand the disease. And we're able to do this method developed in the lab, informally called TIM. Noam just spent a good amount of time explaining this, so I get to save this time for more results. But I did want to quickly just give a reminder. So essentially the key idea with this method, right, is we're utilizing sparse longitudinal data. We run TIM, which then leverages the temporal information that exists with individuals and integrates it across an entire cohort to infer a continuous progression axes. To be clear, this is not pseudo-time. And just to remind about the car analogy, right? So if you think about it, it's like an individual is a car, the timeline of disease is the road, and then we are able to essentially pick up different characteristics of this road, so things like speed of driving. And so with this method, we applied it to this phenomenal data set called PPMI, or Parkinson's Progression Markers Initiative. PPMI is a large multi-center longitudinal cohort. And so, like I said in the beginning of this presentation, this project is still very much a work in progress. So PPMI is made up more than just these data modalities, but these are the ones that we decided to start with. And here in this table, what I'm showing you is the individuals and samples. As you can see, we have PD, prodromal, and healthy controls. RNA-seq is our largest cohort. And essentially samples are taken roughly across 5 years. Great. Okay, so with this data, we're able to ask this question now, and using TIM, can we recover a continuous, biologically meaningful timeline of PD progression from multimodal data? And we found that we can. we can build these timelines. So what I'm showing you here on this plot, the x-axis is the different modalities, are the different timelines with these modalities. The y-axis is tau timeline, which Noam already explained, but essentially is an estimated metric of performance. And as you can see, you know, the higher the value, right, the better. And so we're doing pretty well. And what I wanted to point out here is that one, yes, we're able to build timelines, but also that we're able to build timelines in different tissues and across different data types, which was really nice to see. And so this is all great, but this brings us to our next question, is that whether or not all these timelines are actually underlined by the same road. You know, bringing this back to this car analogy, right? So these are all, you know, on these highways, but are these all, you know, on this one same PD highway? And so in order to answer that question, we just did a correlation of these different inferred timelines. And the idea is that if these are actually representative of this true timeline of PD, that they should be related to each other. And that's what we're seeing here, right? So here you have the different inferred timelines. These are the correlation. This is row values, and the stars denote significance. And so we're doing a pretty decent job, but that's not enough. So what we wanted to see is whether or not these timelines can relate to real clinical presentation of the disease. And so we decided to focus on RNA-seq since it was our better performing timeline. So all the results going forward are in the context of RNA. But I'll also have the RNA-seq a little graphic thing to remind. Okay, so what we did is we correlated the timeline to different metrics related to the disease profile, which you can see here on the x-axis. And as you can see, individuals progressively acquire features expected of more advanced disease states. So essentially, we're finding that we can recapitulate time, which was really exciting. So this is a pretty nice validation. And so the next question that we had, as you can recall, there are lots of symptoms that are related to PD that are, impacting different systems. And so what we wanted to do was to look at the multi-system clinical measures that are associated to be PD, and we started with motor. And so what you see here is we are seeing, for different measures of motor effects, we are seeing significant correlations. So next, we also know that cognition is another important clinical metric of PD. And so as time progresses with Parkinson's, there can be impaired cognition, which has, I think, generally been less characterized in actual progression in the disease, and it can be a little bit less obvious. But when we actually see our associations with the inferred timelines to the measures for cognition, we are seeing correlations, significant correlations, which was great. Next is sleep. So sleep is something that has also been found to be associated with Parkinson's. And generally, I think you see these disturbances earlier in the disease, typically in prodromal periods. And excitingly enough, we're also seeing significant correlations. The next thing that we were interested in to look at is neuropsych, which I'm assuming with this crowd, I don't have to convince is a very interesting and important area to look at. In PD, the different neuropsych effects can really affect the quality of life for an individual. And I don't think we know a whole ton about how it progresses throughout the time course of the disease. And so, where is my slide not changing? There we go. Neuropsych. So yeah, what we see here is a bunch of significant correlations across different measures within, neuropsychological. And yet another system at play in Parkinson's, especially in these earlier periods of the disease, is also autonomic dysfunction. And we see a significant correlation there. And finally, we saw significant correlations when it came to treatment, which, as you can imagine, opens up all kinds of cool questions that we can ask. But yeah, so these were the correlations that we started with. And as you imagine, like these bring up a lot of unanswered questions that we can continue to dig into. But what we wanted to do first, before diving deep into this, was to essentially look at subgroups of PD. So as I mentioned at the beginning of the talk, PD is this very, it's a heterogeneous disease. There's a lot of different ways that it can present. And so there is this hypothesis that maybe there's like sub timelines in this disease. And we wanted to see if we could start, getting at this. And so we decided to start with the fact that there is causal variation that is associated with PD. And so we wanted to see if we could figure out if people with different genetic profiles associated with PD are in the same, highway or road or timeline of PD that we've built. Unfortunately, PPMI has these subcohorts that include these individuals. And so this is just a table showing the amount of individuals and samples. And so essentially what we did is we built timelines for each of these three groups. And we actually found that, they performed quite well. And so here in this figure, you have each panel essentially is one of the timelines that we built, and this is the tau. And you can see we're performing quite well, which honestly was, very exciting. And so the next question that we had is, are these distinct, right? Like, okay, we can build these timelines, but are they actually different from other timelines? And so in order to answer that, what we did is we projected the other groups onto this timeline, where essentially the idea is that if tau is worse, right? So if the performance is worse, it essentially means that these individuals are likely part of a different underlying timeline. And we see that, which again was very exciting. Where here, so in sporadic, for example, we have GBA and LRRK2 that were projected. And as you see, performing worse. GBA and sporadic projected on LRRK2 here, and then LRRK2 and sporadic projected on GBA here. So overall, doing worse, which essentially is telling us that these timelines that we build from these subgroups are actually independent timelines. And so from here, the next question that we had is, okay, we have these timelines, but based on the literature and just, one would imagine that these timelines may have different characteristics, one being differences in actual progression rate. And so that's what we decided to look at, and we found that we do see differences in progression rate. And so what I'm showing you here, this is a comparison between the GBA slope and sporadic, or essentially progression rate between these two timelines. This is the results from a Wilcox test. And so what you're seeing here, what this seems to be telling us is that GBA progression is faster, which actually is supported by the literature, right? So in the literature, it's found to be that actually people that have GBA variants in PD are actually generally progressing faster, which, the fact that our results showed us that was really exciting. And interestingly enough, when we compare LRRK2 and sporadic, we don't see a significant difference, which also based on the literature is expected. So yet again, there's a lot more work to be done here. I feel like with every time we have one of these results, it's like a whole can of worm of more questions to be asked. But hopefully with all of this, I've gotten all of you guys a little bit excited about the really cool questions that we can get at when we're able to study the timeline of PD. And also just the many more questions that need to be understood. And now with, the tools that we have and the incredible data sets that exist, that we can start to try to understand them. And so just to wrap up, there you go. Just some conclusions. So PD timelines, what I've shown you is that PD timelines can be inferred from multiple modalities, that these inferred timelines correlate to disease severity, treatment escalation, and multi-system clinical progression, and that genetic subgroups show distinct timelines and progression rates. As I've said multiple times in this talk, there's so many more questions, so there's a lot more work to be done here. But things that I'm particularly interested in looking at and that we can look at is one, correlating the clinical metrics, the genetic subgroup timelines. So right here, I showed you all these correlations. Oops, sorry. I showed you all these correlations to these different metrics for our original timeline. But it would be interesting to see with these, subgroups or these subgroups timelines that we've created. Can we also correlate them and see differences? Characterize the differences in timeline inferred from different modalities. So at the beginning, I showed you that we have built these timelines across different modalities, but then just further characterizing them and seeing what are the differences, what are the things in common, and what are things that we can learn from each and together. And another thing is building other subgroup timelines. So we focused on genetic subgroups here, but there are other groups that could be looked at that could present independent timelines. And so with that, obviously, acknowledging Noam Beckmann and the Beckmann lab. And I did want to really acknowledge PPMI, especially the participants and investigators. This data set has been very pleasant to work with, and also just an incredible resource that really allows us to ask a lot of questions that we wouldn't be able to ask otherwise. So thank you to everyone that worked on that, and thank you to all of you guys for listening. And with that, I will stop sharing.
Yeah, please everyone, stay for the discussion. These were 3 wonderful talks, so we can start answering some questions from people. I think people can unmute themselves, right? If they want to ask things. But I'll start with the first question for Noam. From Jasmine. She's asking if you expect your timeline-based approach to be applicable to diseases where diagnosis can occur at multiple points through an individual's life, like depression, in order to construct a disease timeline? And if so, what modifications, do you have to do to the method as necessary?
Thank you, Jasmine, for this question. This is a really important question. The short answer is yes. And we actually have data to show that when we take other diseases with different onset, we build, for example, timelines of COVID-19, where people get infected at widely different ages. Some are in their teen years, some are as adulthood, some are late life. And when we build this timeline of COVID-19, they are all on the same timeline. So these timelines are independent of age of onset. And I think that's a really critical point here in the way that they're trained. So I don't think there's many modifications that would need to happen there. And we should absolutely try. We haven't built any MDD timeline. I think it'd be fantastic to do so. And so I'd love to think about that with you if you're interested, and please reach out.
Awesome. We have one more. For Anina, what does pseudotime mean? If you can define that.
I think he ended up dropping the question afterwards.
Oh,
But essentially, from my understanding, essentially it's like pseudotimes more like the actual biological process, like it's essentially inferred more from biological processes, whereas we're actually picking up on like actual time, like real time. So yeah.
I can ask a question.
yes.
Since there's nothing pressing in the chat, I would love to take advantage of having Hongtu and Noam here, because I feel like your results are could be interpreted as a little bit at odds. So Hongtu, you have this genetic correlation near unity between your time to event, if I understood correctly, between your time to event traits and the risk phenotypes. But Noam, you're saying that the genetic architecture of AD progression is distinct from the genetic architecture of AD risk. These are, genetically two different things. And so I wonder if, maybe one at a time you could each explore that, what I perceive to be a tension.
I'm happy to start by saying I don't think there's any tension there.
Okay.
The way I understand what Hongtu has presented is fundamentally consistent with what we know about Alzheimer's. Age, which I would define as an environmental factor, obviously is important. As you get older, your risk increase. If you're young, unless you have some form of early onset Alzheimer's, you're not going to have Alzheimer's disease. You're not going to have any cognitive deficits due to that. So I don't think they're mutually exclusive at all. I think if anything, they're complementary. And I just want to also emphasize that what we're studying here is not time of onset, rather progression rate. And these are also fundamentally different concepts. So again, in my opinion, they're not exclusive, they're complementary. And I'm sure we can do even better by integrating them together.
Thanks.
Yeah, I agree. I think in generally, and for many of the diseases, it's not a simply case controls, because that's type of information and does not really delineate how the disease progress. And for many of the diseases, and basically the like Alzheimer's disease, when you're getting older, the chance of you getting Alzheimer's disease is getting higher. And I think that's the basic additional information you can basically it's important for many important biological questions.
Thank you.
I also have one for Hongtu as well. Does the model differ if there is a specific gene that really influences the later stages of the disease compared to having an early onset? So, is this affecting how the model performs?
I'm not quite getting with your questions.
Yeah, so what happens, if a gene only impacts the later stages of a disease rather than, speeding it up from the very beginning? Is this affecting how the method performs, the assumptions?
Yeah, I think this is because our phenotype is the survival time, and basically when this basically the disease happened. if a gene genetic markers could influence basically later of the survival, the transitions. Yeah, I think that this is also related to the study designs because then when you could have, yeah, you could have sensor because for the subjects have that genetic markers, you high chance they get the data will get censored because you typically you stop a study, you know, and I think they could, but I think for our method is even you see that for UK Biobank data is highly censored. And I think our results even for sensor rate is 99% is works reasonable well.
Okay, we have one more question we can definitely answer live before wrapping up. So how would you integrate the comorbidities over the lifespan? For example, in neurodevelopmental disorders that the onset is early, however, phenotypes and comorbidities are varied. So I think this goes for all of you possibly, right? As a question. So whoever would like to answer that.
I'm also happy to start here. I think this is a critical question. I think that extends to not just neurodevelopmental disorders, but really everything out there. We always have to think about comorbidities, etc. In the context of timelines, one of my dreams, which may or may not happen depending on if funding follows on that, is what if we could actually start building these timelines for every disease? Because at the end of the day, every single comorbidity is a different disease. And then what we can start thinking about, if you again go back to the car and road analogy, the goal here is really to start defining an atlas of all these timelines and how they interact with each other and how they impact each other. And I think that's how we're going to better understand disease progression in general and hopefully resolve a lot of the problems that are still around for human health.
Any other thoughts?
I mean, I'll just echo Noam, just that I think there is, especially like Edna, your point, it was just like neurodevelopmental disorders, like yeah, like things happen earlier, and so being able to actually like look at those things earlier and actually like piece and characterize these timelines, as Noam's point, like these atlases and like being able to see everything, it's really just going to give us a clearer understanding of like what is happening when, and then and being able to tie those things later on. So it's a phenomenal question.
In the interest of time, and I'm not seeing too many other questions popping up, I will thank our speakers again for sharing their research with us today, and thank you all for attending. And this webinar will be available on YouTube, the recording of this webinar, and I hope you all have a wonderful day.
Thank you.
Thank you.
Thanks, everyone.